Hi, and welcome to this webinar about newly launched Alpha CETSA kits. My name is Nancy Decky, and I am working as a senior research scientist here at Pelago Bioscience, and I'm going to lead you through how the CDK2 and CDK4 Alpha SureFire kits have been validated for CETSA and how they can be utilized for high-throughput screening. Pelago Bioscience has been using the Alpha technology from the start of the CETSA HT format and the usage of plate-based assays. As Pelago holds the expertise of the CETSA technology and PerkinElmer have the expertise of regions and detection technologies, we entered into an Alpha CETSA collaboration in 2018. The CETSA technology is treat, heat, detect, and then navigate through your chemical libraries. The advantages of CETSA is that it can be made in high throughput format with the dual antibody detection system, which is based on proximity. To study target engagement with CETSA, the compounds of interest can remain unmodified and no tags are needed. The target of interest is preferably endogenously expressed and doesn't require a label. Since the technology is used in a natural environment, the CETSA technology is of high physiological relevance. Since last year, Pelago has CETSA validated 9 targets from existing Alpha kits. The first launch included 5 assay kits, which were presented at the SLAS conference in Barcelona last year. 3 more kits were launched in autumn the same year, and 1 additional important target was added to the list this year in January. So different target proteins have been selected at this first hand to show the diversity of the Alpha CETSA technology. And that is that CETSA can be applied on many different protein families, cytosolic and membrane proteins, as well as nuclear target proteins. The list of CETSA validated Alpha kits are expanding, and now we are launching 2 members of the CDK family, the CDK2 and CDK4. These assay kits are based on the Alpha SureFire technology and developed by TGR Biosciences, who are in close collaboration with PerkinElmer and Pelago Bioscience. CETSA is based on thermal stabilization of proteins upon ligand binding. It reveals direct target engagement with a label-free assay in living cells. When a native folded protein undergoes a temperature gradient treatment, it will denature at higher temperatures. And when applying a drug, the melting of the target protein changes, and there will be a thermal shift. What is detected here is the amount of folded protein that is left after heating at the different temperatures. The detection of folded proteins is done by the Alpha technology, which is based on proximity. So when the protein is folded, the target antibodies will bind and make the donor and acceptor read in close proximity, and an energy transfer will occur. In contrast, when the target protein has been heated to higher temperatures, the target antibodies are not in close proximity, and an energy transfer will not occur, and you will have no signal. To do a validation, a tool compound is required to be able to determine if the target protein can be shifted. The compound criteria for CETSA are that the compound should bind directly to the target. It is important that it has high solubility, since a high concentration of the compound is used in order to saturate the system to verify target engagement. To study and validate the target engagement of commercially available tool compounds for CDK2 and CDK4, we included several classes of inhibitors such as covalent, active, site, and allosteric binders. This is to highlight the versatility of the technology. To apply the CETSA technology for a target in an existing Alpha kit, the target is validated in 4 steps: analyte titration, lysis buffer selection, melt and shift curves analysis, and XC50 determination. In the first step of CETSA validation, we titrate the cell number that is required to have a good signal to background. 10 serial dilutions of the biological samples are made, and 5 is heated to 37 degrees to detect the maximum protein levels in the cells, and 5 is heated to 70 degrees to detect the minimal protein levels in the cells. After heating, 5 lysis buffers that are formulated and also available at PerkinElmer are applied, 1 to each dilution within the group of 5. The cell lysates are then added to the alpha detection plates and detection consumables are added. To the right, the selected lysis buffer is indicated for the CDK2 and CDK4. For CDK2, lysis buffer number 3 and CDK4, lysis buffer number 4 was selected. There is a high signal to background in all cell concentrations, but preferable is to have as low cell number as possible in the final detection plate. So for CDK2 and CDK4, 6,000 cells per well was selected for further validation. The cell number and lysis buffer are important parameters to perform melt and shift curve analysis with a good signal to background. In the third step of validation, the selected 2 compounds are incubated with intact cells and heated in a 12-step temperature gradient. The cell compound suspension is then lysed with the selected lysis buffer and then transferred to an alpha detection plate where the CDK2 and CDK4 detection consumables are added. Detection is made with an alpha-compatible plate reader like a Victor Nevo or an Envision, and the following data was collected with an Envision. The CDK2 and CDK4, 2 compounds, shifted the melting temperatures of the target to the right, which makes them categorized as stabilizing compounds. From the melt and shift curves, a temperature can be selected to perform EC50 determination, where the cells are incubated with serial diluted compounds and heated at one single temperature. In this case, 58 degrees for CDK2 and 46 degrees for CDK4 were selected. The concentration response curves at the indicated temperatures is shown at the bottom of this slide, and the CETSA EC50 values are in the micromolar range. When the compounds are binding in the active site in the ATP binding pocket, the compound may not be able to compete out ATP and be able to bind. To study these compounds, the cell matrices need to be diluted out from ATP. So the solution is to use disrupted cells that are incubated with the compound. And this was done to detect target engagement with 2 compounds for CDK2 that wasn't able to compete out ATP in an intact cell environment. But in a disrupted cell environment, both compounds were able to bind to the target and shift the melting of the protein to the right. And we could then determine an EC50 value at an isotemperature of 55. A pre-validation for high-throughput screening has been done with the CDK4 alpha CETSA kit here at Pelago, where we used a pre-spotted DMSO plate for cell incubation, for which an isotemperature of 46 degrees was applied from column 1 to 22. Controls were added and they were heated to 37 in column 23 and 70 degrees in column 24. A very even alpha signal distribution was obtained from column 1 to 22, and the signal to background was very good, looking at column 23 to 24. The assay showed good statistics with an acceptable set prime and a signal to background. Therefore, the CDK4 alpha SureFire CETSA kit is a very well suitable for detection and usage in high-throughput screening. Target engagement is a measure of compound binding and can be determined in a natural environment with the alpha CETSA technology. It can be applied at several stages in drug discovery, primarily during high-throughput screening and hit confirmation, as well as in lead optimization for SAR analysis. Finally, I would like to acknowledge our collaborators Perkin-Elmer and TGR Biosciences for the work that we have done together during this validation process, and especially Nicolas Pierre at Perkin-Elmer and Anthony Sheehan at TGR Biosciences. For any questions or support regarding the alpha technology, SureFire kit development, or the CETSA technology, please use the contact details indicated on this slide. Thank you so much for listening.